Hi everyone, this is Reema, your biology educator from VLearning. So as you know that VLearning is an online educational platform where we strive to provide quality education for all. So to get all the benefits from our channel, do subscribe to it, and also don't forget to visit to our website, that is www.v-learning.in, where you will be getting all the PDF notes of these topics. So without any delay, let's begin today's session. So in the previous class, we have started with your chapter, right? So we have started with chapter number 8, isn't it? So we have looked into the overview of the cell, so where you have understood about who discovered cell, right? And we have looked into the various cell organelles present inside the cell, and also we have you have learned about that what are the two types of cell, like prokaryotic cell and the eukaryotic cell. And we also have discussed about what is the difference between the plant cell and the animal cell, right? So in today's class, we are going to learn in detail about the prokaryotic cell, and this is your lecture number 2. So what do you mean by this prokaryotic cell? As we already discussed in the last class that prokaryotes, right? Prokaryotes means these are the primitive cell, right? That is the bacteria, cyanobacteria, all this comes under the prokaryotes. So prokaryotic cell means, what does pro means? Pro means the primitive, right? Pro means primitive. So pro means primitive, and karyon means what? Karyon means nucleus, right? So they have what? They have the primitive nucleus. So as you can see, this is the picture of the prokaryotic cell, that is the bacterial cell. So when you look into the bacterial cell, it will give you a picture of this. You can see it properly with the help of the electron microscope. We cannot see this prokaryotic cell with the naked eyes. The bacteria which we cannot see with our naked eyes, we can see it only with the help of microscope. So when you look into the structure, ultrastructure, you will see that the prokaryotes, they are having the circular DNA. And also in the prokaryotes, you will not find the cell organelles. Okay, most of the cell organelles are absent, as we have learned in the previous class, right? That most of the cell organelles are absent in case of the prokaryotes. So what we find, only the ribosomes, right? We find only the ribosomes. So you can see this dots like structure, right? What are this? These are the ribosomes. Okay, so we find only ribosome. And in case of the prokaryotes, the ribosome is what? The ribosome is 70S. Okay, the ribosome is 70S. So what are the various structure of the bacteria? You will find this pili. Okay, in case of bacteria, you will find the pili. This pili helps in conjugation. You will find the DNA. They have the genetic material. This is nothing but the DNA, which is the genetic material, and their DNA is circular. Okay, their DNA is circular. And besides the DNA, you will also find the plasmids. Okay, these plasmids are also present in the prokaryotic cell. So what are these plasmids? These plasmids are nothing but the extra chromosomal circular DNA, which is also present in case of the bacterial cell. And you can see this blue color, right? So this is the fluid matrix. We call this part as cytoplasm. Okay, so this is called as cytoplasm. Now talking about this prokaryotes, that is the bacteria, these are the smallest cell. Okay, so these are what? These are the smallest cell. These are the smallest cell. So as we know that mycoplasma is the smallest cell, and yes, the bacteria are the smallest cell, and which we cannot see with our naked eyes. So they will be present everywhere inside our body. Also they might be present. There are some bacteria, which may be good for us, and some are very much harmful for our body. They cause lots of diseases. Okay, so many diseases because of this bacterial infection. Now, talking about the body of this bacteria, do they have cell wall like the prokaryotes? They also have what? They also have cell wall. So these prokaryotes have what? They have the cell wall. The cell wall is present in case of the prokaryotes. Okay. So next, let us learn the various characteristics of this prokaryotic cell. So this prokaryotic cell, as you know, that they are the smallest cell, right? And also they have the capacity to multiply at a very, very faster rate. Okay. So they follow which mode of reproduction? Yes, they will be following the asexual mode of reproduction. Okay, as you have learned in the last class that all the unicellular organisms, they follow what? Asexual mode of reproduction. So look into the characteristics of the prokaryotic cell. As you can see that they are generally what? They are generally smaller and multiply more rapidly than the eukaryotic cells. So they are the smallest cell, right? And they have the capacity to multiply more rapidly. So examples are the bacteria, blue-green algae, then the mycoplasma and the PPLO, that is the pneumonia-like organisms. Okay, so these are the examples of what? These are the examples of your prokaryotic cell. Okay, and they vary greatly in shape and size. So the size of the bacteria, the size of the prokaryotic cell, it may differ from one another. That is, the size varies. All right. So next, yes. Let's see the shape of the bacteria. When we talk about bacteria, this bacteria again have got different shapes. Okay, they may be round, they may be rod shaped, they may be spiral, or some of the bacteria may be coma shaped. Okay. So this bacteria, depending on the shape of the bacteria, they are again classified into 4 types. Okay, so what are the 4 types? That is the bacillus. Okay, bacillus is what? Bacillus is a rod shaped bacteria. Okay, how it looks like? It looks like this rods. Okay, so it is a rod shaped bacteria. And this coccus is spherical or oval in shape, so it looks like this. Okay, it is spherical and oval in shape. So yes, when many coccus they combine together, okay, we call it a streptococcus. Okay, streptococcus. And vibrio, so how it looks like? It looks like coma shape. Okay, and the spirillums, it looks like spiral shape, so it is spiral in shape. So these are the various shapes of the bacteria. And these bacteria are responsible for causing harmful diseases in case of human beings. And some of the bacteria, like when we are talking about the bacillus, that is lactobacillus. Lactobacillus is one of the bacteria which is very much helpful in the process of fermentation, right? So this is a useful bacteria which is used in the process of making the curd, yogurt and all. So this is a good bacteria. So next, you can see the picture of the bacteria. When you see in the electron microscope, you will get a view of this. Okay, so this is the picture of the bacteria. You can see here the various cilia and the flagella, what they have. Yes, some more characteristics of the bacteria. As I mentioned that this bacteria have what? They have the cell wall. Okay, they have a cell wall. Like in case of plants, they have cell wall, right? So this bacteria also have a cell wall surrounding the membrane except in mycoplasma. So this is the bacteria, that is the mycoplasma, which is known as the smallest bacteria. So it measures about 0.3 micrometer. Okay, mycoplasma, it is the smallest bacteria. So in case of this mycoplasma only, the cell wall is absent. Clear? So they have a cell wall. So when I talk about the cell wall, what do you mean by this? Cell wall is what? It is the outer membrane. It is present just after the plasma membrane. So if this is the plasma membrane, so see, if this is the plasma membrane, so the cell wall will be, the cell wall will be which one? The cell wall will be this one. Okay, this is the cell wall. Alright, so this is the cell wall. The cell wall is surrounding just after the plasma membrane. And what is the function of the cell wall? The cell wall is providing support a rigidity to the bacterial cell. All right, so the cell wall of the bacteria is made up of your polysaccharides or the murin. Okay, so it is providing protection. Okay, it is providing protection and support to the bacteria. So next is the fluid matrix filled in the cell is called the cytoplasm. So as I have showed in the picture that this area where the cell organelles lies, okay, this is the DNA, okay, then these are the ribosomes, right? This area that is the fluid matrix where all the cell organelles are lying, we call this place as cytoplasm. Okay, cytoplasm. So basically cytoplasm is also responsible for the transfer of the food materials. It acts as a junction, okay, and it is providing the support. It acts as a matrix for all the cell organelles. Clear? Yes. There is no well-defined nucleus. So in case of the prokaryotes, they do not have well-defined nucleus. Okay, so as you can see that the nucleus is not properly formed. So the DNA is lying in the free surface. Okay, the DNA is lying in the free surface. The nucleus is poorly developed in case of them. Okay, the nucleus is not properly developed, and the genetic material is naked. That is not enveloped by the nuclear membrane. So you can see that the DNA is naked DNA, right? And it is not covered by the nuclear membrane, isn't it? So in case of them, they do not have nuclear membrane, and also the cell organelles like mitochondria and the plasmic reticulum, Golgi bodies, all these are absent in case of the prokaryotic cell. Clear? So only what they have the ribosomes, right? So these are what? These are the ribosomes, and what they have the inclusion bodies. Okay, these inclusion bodies are responsible for the storage of the food. Okay, so they have inclusion bodies. So this is all about it, and many bacteria have a small circular DNA outside the genomic DNA, and this smaller DNA are called the plasmids. So as I said that, so this is the genomic DNA, right? So outside the genomic DNA, they will be having a circular genetic material. Okay, so what are this known as? These are known as the plasmid. Okay, so these are known as plasmid. So what is this plasmid? This plasmid is the extra chromosomal circular DNA that has the capacity to self-replicate. Okay, so what is this plasmid? Plasmid is the extra chromosomal circular self-replicating DNA. Okay, so it is also present in case of the bacterial cell. So this plasmid, it also acts as a vector. Okay, next. Yeah, so let us talk about the structure of the prokaryotic cell. As you have already seen that what are the things present. So if I draw the diagram of the prokaryotic cell, so it will be having a capsule. Okay, the capsule is present just after the cell wall. So like this, it will be having the flagella, long flagella it has. Okay, this flagella, it helps in the process of movement. So it has got a capsule or the slime layer. Then what else it has? It has this cell wall, right? And it has what? It has the plasma membrane. So see, so this is what this is, the plasma membrane. Then on top of the plasma membrane, what it will have? If this is the plasma membrane, so the top layer on top of the plasma membrane, it has what? It has the cell wall, right? So this is the cell wall. So next, what it will be having? It contains the genetic material or not? Let me take this red color. So it will be containing genetic material. The genetic material is lying in the free space, and it looks like a wool, right? So it is tangled. Okay, it is not properly covered with the help of the nuclear membrane. Clear? So this is the genetic material which lies in the free space in the cytoplasm, and also it will be having what? It will be having the ribosomes which will be lying in the cytoplasm, right? So it has the ribosomes. Then what else it has? It has what? It has the inclusion bodies, okay? So these are the various inclusion bodies this bacteria has. Then in the body of the bacteria, you will see like this structures, that these are known as pili, okay? So these are known as pili, okay? The pili helps in what? The pili helps in the process of conjugation. So see, they will be having pili, okay? Somewhere you will be finding the cilia too, okay? In between the pili, you will find the cilia also. The cilia helps what? The cilia helps in the process of movement, okay? Locomotion. So yes, all the body is covered with pili and the cilia, alright? So what is this? This is called as pili, okay? This is called as pili, not R, but P, okay? So yes, pili, right? Then this is known as, this structures are known as what? The structures are known as cilia, all right? The cilia helps in the process of movement. This is what, this long structure is known as the flagella, okay? This is known as flagella. This is how the bacteria's structure looks like, okay? So inside, what we are finding inside, you will find the DNA, right? This is the genomic DNA, okay? So they have got a circular DNA and it is not covered with a nuclear membrane, right? And also what this black dots, what you can see is what? This is called as the inclusion bodies, okay? These are called as inclusion bodies, right? So what is the function of this inclusion bodies? They are basically responsible for storage of food, okay? So this inclusion bodies are responsible for the storage of food. And this is what, this is flagella, right? This is helping in the process of movement. Some of the bacteria are motile, so they use the flagella for the movement, right? And it has got a capsule. It provides them extra protection, and as you can see, this dots like structure, these are what? These are the ribosomes, all right? So these are the ribosomes. So this is how the bacterial structure looks like. In case of bacteria, they have got 70S ribosome, and this ribosome is responsible for the process of protein synthesis, all right? So what they do? They are responsible for the process of protein synthesis. They help in the process of protein synthesis. So this is the structure of the bacteria. So we will be discussing in detail about the various parts of the bacteria in the next topic, okay? So see, the first one, the first part talking of the structure of the bacteria has got the cell envelope, and its cell envelope consisting of three things, okay? So what are the three things present in the cell envelope? The first thing is it has the glycocalyx, okay? It has glycocalyx. Next thing is it has the cell wall, and the third one is it has the plasma membrane, okay? It has plasma membrane. So all these three components, it makes up the cell envelope of the bacterial cell, okay? Now talking about this glycocalyx, cell wall and plasma membrane, how they are arranged. So if this is the cell of the prokaryote or the bacteria, so the plasma membrane will be this one, right? The plasma membrane will be this one, which is present inside layer is the plasma membrane, right? Then on top of the plasma membrane will be the cell wall, isn't it? Then above the cell wall, they have got extra covering, okay, extra protection. So as you know that they are very, very small, so they need extra protection. So that is known as what? That is known as the glycocalyx, okay? That is known as glycocalyx. calyx. So this glycocalyx, it can be made up of capsule or it can be made up of slime layer. Okay, it can be in the form of capsule or slime layer. So see here, bacteria cells have a chemically complex cell envelope. Okay, they have a complex cell envelope which provides them what? Which provides them protection. Okay, which provides them protection from the damage. Okay, and the cell envelope consists of a tightly bound layered structure, that is the outermost glycocalyx. Okay, the outermost layer is which one? The outermost layer is the glycocalyx, right? After the glycocalyx, what is present? Cell wall, isn't it? Then next is the plasma membrane, right? So see, followed by the cell wall, right? And then the plasma membrane. So these are the three things which is making up the cell envelope. So it is together, it makes up the cell envelope. Clear? So this is how the cell envelope is arranged in case of the bacterial cell. So let's look into the glycocalyx. Okay, so the glycocalyx, as I have said, that it will be thick. Okay, and the composition of the thickness will differ from bacteria to bacteria. So see, some of the bacteria can have a glycocalyx which is of loose sheet. Okay, so that is known as the slime layer. Okay, that is known as slime layer. And some may be having a thicker glycocalyx, that is known as capsule. Okay, so that is known as capsule. So again, the glycocalyx is divided into what? The glycocalyx is divided into two parts, right? One is what? One is the capsule, isn't it? One is capsule, another is what? Another is the slime layer, right? So this capsule and slime layer, what it does, it helps the bacteria to attach inside the body. When any of the bacteria, for example, it is causing harm to your intestine, right? So it needs to reach to our intestine and it will attach to the cells of the intestine, isn't it? And it will be destroying your immune system. It will weaken the immune system, right? So it helps the bacteria to attach to the cells, that is to adhere to the cells. Okay, to adhere to the cells of the host. So this capsule and slime layer, what it does, it provides adherence. So this capsule is having the tough layer. Okay, so it is that having the thickness, it is thick and it is tough, right? It is tough. And this slime layer, it is not having much thickness. Okay, it is not much tough. Okay, it is loose. Okay, it is loose, but it also helps the bacteria in adherence. Okay, adhere to the cell. So this is the function of this capsule and the slime layer. So it is the outermost layer consists of a what? Coating of polysaccharide or the murine. Okay, so this glycocalyx, it consists of what? It consists of the polysaccharide. It consists of polysaccharide or murine. Okay, so it is having this composition. Okay, polysaccharide or murine is present in a glycocalyx, and what it does, it helps in adhesion. Okay, it helps in adhesion or adherence. So this is about glycocalyx. Now let us look into the cell wall. Okay, so what is the function of the cell wall? You know that cell wall is responsible for providing support and protection to the cell, isn't it? So in case of this bacteria, we can identify the bacteria depending on the cell wall. All right. So some of the bacteria will be having a thick cell wall, and whereas some will be having thin cell wall, right? So we can identify the bacteria based on the cell wall. So based on the cell wall, when the staining was done, okay, what is the staining? Staining is nothing but it is a process, okay, where we can identify the bacteria with the help of using these chemicals, okay, in the laboratory, okay. So it is a process where we can identify the bacteria through the staining. So depending on the cell wall, the bacteria is classified into two types. One is gram-positive and another one is gram-negative. Okay, so one is gram-positive and gram-negative. So these are the two types of bacteria depending on the depending on the cell wall. So this gram-positive bacteria, they have got thick cell wall. Okay, they have got thick cell wall, and this gram-negative bacteria, they have got thin cell wall. So when the staining was done, this gram-positive bacteria, it will be identified as purple in color. Okay, when the staining was done, after staining we can identify the bacteria as this purple color. So this purple color represented what? Gram positive bacteria, whereas this gram negative was represented as pink color. Okay, we will learn how the staining is done. Okay, so see, on the basis of cell wall, it is classified into gram positive bacteria and gram negative bacteria, right? Now let us see that how the staining is done, or who is the scientist who has first discovered the staining process. Okay, it is done basically in the laboratory. Okay, so one of the scientists, famous scientist who has discovered this technique, his name is known as, or he is known as Christian Gram. Okay, Christian Gram. So he is the first person who is who has done this staining process in order to identify the bacteria. Now, how he did this staining process? For that, what you need for staining, you need some of the chemicals. Okay, some of the chemicals like safranin, then alcohol, then iodine. Okay, these are the requirements. So as you can see in this picture, right? So these are the various colonies of the bacteria it is being formed. But how it is being done? It is done in the in vitro condition. Okay, that is inside the lab. We need proper condition. Okay, and we need to maintain the proper temperature, light, in order to prevent the contamination. So see, these are the various colonies, and you can see this petri dishes. Okay, the bacterial colonies are basically grown in the petri dish. Okay, so inside the lab we have to use a matrix where we can grow the bacterial colonies. So generally the agar is used as a matrix for the culture of the bacterial colonies. So how the staining is being done is, for the process of the staining, the scientist has used this crystal violet. Okay, you can remember like this, crystal violet. Then he has used the iodine. Okay, and he has used ethanol, and he has used what? Safranin. Okay, CIES. So C represents the crystal violet stain. Okay, crystal violet. Then I represents iodine solution. Then E represents the ethanol, which is the alcohol we need for washing it. And S represents the safranin. Okay, S represents safranin. All right, so these are the 4 things which is required for the process of this gram staining. One is what? One is the crystal violet stain, then which will retain the purple color. This will be purple in color. Okay, then iodine. Okay, then ethanol, and then safranin. These are the 4 things which he has used for the process of staining the bacteria. All right. So it is used to classify the bacteria based on the cell wall. Okay, now we have talked about the cell wall, and you know that gram positive bacteria will be having thick cell wall, isn't it? And gram negative will be having the thin cell wall. So based on the property of the cell wall, this bacteria can retain the stain. Some of the bacteria can retain the stain, whereas some will not. Okay, so those bacteria that take up the gram stain, okay, that is the gram stain, are called the gram positive bacteria, and others which do not take up the gram stain are known as gram negative bacteria. So the bacteria which got stained with the help of the staining solution, okay, we call it as gram-positive bacteria. Whereas the bacteria which didn't take the stain, the bacteria which does not take the stain, we will call it as gram-negative bacteria. Clear? So yes. So what is the difference between this gram-positive bacteria and gram-negative bacteria? As you can see, they have got thick cell wall, right? And this gram-negative bacteria, they will be having the thin cell wall, isn't it? Then they have got a periplasmic space present between them, okay? Then they also have this periplasmic space, and then lipids are what? Lipids are absent or very small. Whereas in case of gram-negative bacteria, they have got a liposaccharide layer. That is, lipids are present, okay, in case of the gram-negative bacteria. And teichoic acids is present in case of gram-positive, whereas in case of it, it is absent. Clear? And peptidoglycan can be 16 to 18 nanometer. Okay, what is the size of peptidoglycan? Peptidoglycan is 16 to 18 nanometer. Okay, and whereas in case of this peptidoglycan is what? 2 nanometer. Is that clear? So it is 2 nanometer. So yes, let us see how the staining is being done. For the process of staining, what we need the petri dish, right? So we need the petri dish. Then what we need the bacterial culture. Okay, we need the bacterial culture which we are going to use, right? Then what happens in the process of staining? We have to use what? We have to use the crystal violet stain. Okay, so this crystal violet, it gives the purple color. Okay, it gives the purple color or the violet color. So after staining the bacteria, first we have to stain the bacteria. So you have to take a slide. Okay, you have to take the bacterial culture. Okay, so next what you have to do, you have to use this crystal violet stain. Okay, so we'll be staining it and we'll be keeping it. Then after that we have to use the iodine solution. Okay, you have to use the iodine solution. So next what you need, you have to use the iodine solution and keep it for 30 seconds. Okay, then after that you have to wash it again with the ethanol. Okay, with the ethanol. So after washing it with the ethanol, then you have to use this safranin solution. Okay, safranin solution at the last. Now what happens is, in case of the gram-positive bacteria, so if this is the gram-positive bacteria, they will be having what? They'll be having the plasma membrane, right? That they will be having the layer that is known as the peptidoglycan layer on top of the cell wall. Okay, so this layer is known as what? This layer is known as peptidoglycan, right? So they will be having this peptidoglycan. So here we are going to stain it with the help of this crystal violet stain, right? So this has the capacity to retain the stain, as there is no pores. Okay, as there is no pore or porine present. Okay, there is absence of lipids, right? So they have the capacity to what? Capacity to retain the crystal violet stain. Okay, crystal violet stain. So because of their thick cell wall, as there is no pore present between, so this has the capacity to retain the stain. So it will appear as purple in color or violet in color. So this gram-positive bacteria will appear as what? Will appear as purple in color. Okay, the bacteria which will appear as purple in color, we call it as gram-positive bacteria, right? So but whereas in case of gram-negative bacteria, what happens? In case of gram-negative bacteria, so in case of gram-negative bacteria, along with the peptidoglycan, they also have another layer that is known as the lipopolysaccharides. Okay, so they have got the lipopolysaccharides. So they have got a lipopolysaccharide layer along with this peptidoglycan, okay, along with the peptidoglycan. So here in the lipopolysaccharide layer, they have got some pores, okay, they have got some pores in the lipopolysaccharide layer. So they have got some porins present here, okay. So because of this porins, there is a gap, okay. So there is a gap because of this porins. So because of this porine, there will be gap, okay. So the stain is not properly retained. So when it is stained with the crystal violet, because of this presence of porine, the stain will not be retained, okay. So it will appear as what? It will appear as pink in color, okay. This bacteria will appear as pink in color, that is the gram negative bacteria. So this is how the staining is done, okay. Because of the presence of lipopolysaccharides in case of the gram negative bacteria, they will not retain the stain, okay. They cannot, that is, they do not have the capacity to retain this crystal violet. They will be washed off, okay, when we are going to wash it with the help of ethanol, okay. So they will appear as pink in color, all right. So this bacteria are known as gram negative bacteria. So yes, next let us learn about the plasma membrane, which is another part of the bacteria, right? We have seen the cell envelope, isn't it? So next comes the plasma membrane. So the plasma membrane is what? The plasma membrane is a living cell, and it is very much selectively permeable, okay. So it will not allow all the materials to come inside the cell. It selects, okay, only the material which is suitable to the cell it will allow, okay. So in case of the bacteria in their plasma membrane, so if this is the, okay, if this is the cell wall, right? So if this is a cell wall, then this is the plasma membrane, isn't it? So this is the plasma membrane. So they have got spatial structures, okay. These are known as the mesosomes, okay. They have got like this special structures in the plasma membrane. So they are called what? They are called as mesosomes, okay, mesosomes. So these are the finger-like projections, okay. So this mesosomes help in the process of ATP formation in case of the bacteria. So in case of bacteria, their plasma membrane is having a special invasions, okay. So these are known as what? These are known as mesosomes, and yes, this is what? This is the plasma membrane, right? So this mesosome is responsible for the process of ATP synthesis in case of this bacterial plasma membrane, all right. So this membrane is a similar structure to that of the eukaryote. So plasma membrane, it is also having the same structure like the eukaryotic cell. So a spatial membranous structure is the mesosome, which I have said that mesosome is a special structure which is present in case of this prokaryotes, okay, and which is formed by the extension of plasma membrane into the cell wall. So this is how the plasma membrane is arranged, okay, and these extensions are in the form of vesicles and tubules and lamella, and they help in the cell wall formation. So what is the function of this mesosome? This mesosomes also helps in the formation of this cell wall, okay, formation of the cell wall, then the DNA replication, and also it is responsible, that is distribution to the daughter cells, okay. So DNA replication and distribution to the daughter cells. So these are the functions of what? These are the functions of the mesosomes, okay. It also helps in respiration, secretion process, and also increases the surface of the plasma membrane and the enzymatic content. So these are what? These are all the functions of what? These are all the functions of mesosomes, okay, functions of mesosomes. So you can write it down. These are all the functions of mesosome. So in case of some prokaryotes like cyanobacteria, there are other membranes or membranous extensions in the cytoplasm, we call it as chromatophores, okay, which contain the pigments. So in case of some cyanobacteria, they will be having the pigments that is known as the chromatophores, okay, so which is responsible for the process of photosynthesis. Yes, so as we have already seen the structure of bacteria, so they will be having like this structure, right? So that is known as what? That is known as the flagella, isn't it? So this is what? Flagella. So this flagella is again divided into three parts, that is the hook, okay, right? That is the hook filament and the hook filament and the basal body, right? And the basal body. So these are the three parts of the flagella. So flagella is divided into three parts. It has got hook, filament and the basal body, which helps in the process of locomotion, okay. So the bacterial cell, it can be motile or non-motile, okay. Some of the bacteria will be motile, some of the bacteria will be non-motile, that is it cannot move. Clear? So it has got a thin filament. This thin filament is known as what? Flagella. So as you can see, this is the flagella, right? This one is the flagella, which is the thin filament, okay. And what bacteria shows a range of range in the number and the arrangement of flagella. So it also differs from bacteria to bacteria, okay. So it consists of three parts. Flagella has got three parts. What are the three parts? The filament, the hook and the basal body. Filament, hook and basal body are the three parts, and flagella is that is the filament is the longest part, okay. It is the longest portion and extends from the surface to the outside, okay. The filament is the longest part of the flagella. So beside the flagella, the bacteria also will be having the pili, okay. So as you have seen that in the structure of bacteria, they also have this pili, isn't it? So what is this pili? This pili basically helps in the process of conjugation, okay. They helps in the process of conjugation, that is the reproduction, that is in the process of asexual reproduction. And also they have got fumarae, okay, so which is helping or the surface structures to the bacteria, and they do not play a role in motility. It is only providing surface structure to the bacteria, okay. It is only giving the structure, proper structure to the bacteria, but it does not help in the process of movement. Clear? So the pili are what? They are elongated tubular structures made of special proteins, okay. So they have got one special proteins, okay, and they are the elongated structures, right. And the fumarae are what? The fumarae are the small bristle-like fibers sprouting out of the cell, and in some bacteria they are known to help attach the bacteria to rocks and streams and also in the host tissues. So this fumarae, you know what it does? It helps the bacteria to attach inside our body. When any bacteria attacks the body, with the help of this fumarae, the bacteria can attach to the various cells of the human body or in case of plants, okay. So this basically helps in attachment, okay. And yes, so in case of bacteria, you know that they have only ribosomes, right? So how are the ribosomes of the bacteria? So in case of the bacteria, they have got ribosomes, isn't it? So this dots like structure which we find inside the bacteria, these are what? These are the ribosomes, right? So these ribosomes are what? These ribosomes are responsible for the process of protein synthesis, okay, for the process of protein synthesis. And they have got what? They have got the 70S ribosomes, right? So that is, the ribosomes is divided into 50S and 30S, okay, that is the subdivision, 50S and 30S. Now, how this protein is being formed? Protein is being formed from the DNA, okay? From the DNA, it will be forming what? It will be forming the RNA, okay? From the RNA, what happens? The mRNA will be formed, and from the mRNA, the protein will be formed, all right? So this is how basically the replication, transcription and translation you'll be studying in class 12 standard, okay? So how the protein formation takes place, okay? So this is how, that is from the mRNA, the protein is being formed by the process of translation, okay? So yes, so in prokaryotes, ribosomes are associated with the plasma membrane of the cell. So it will be present nearby to the plasma membrane, and they are about 15 nanometer to 20 nanometer in size. So what is the size of the ribosome? The size of the ribosome will be 15 nanometer to 20 nanometer, okay? 15 nanometer to 20 nanometer, and there are two subunits which you have seen, that 50S and 30S, okay? That is 50S and 30S. 50 is the larger subunit and the 30S is the smallest subunit, okay? So the ribosomes are what? They are the site for protein synthesis. As I have said that the ribosome is responsible for the synthesis of the protein. Clear? So how the protein is synthesized? Through the process of translation, okay? So ribosome helps in the synthesis of protein. So several ribosomes, they attach to a single mRNA form a chain called polyribosomes. So what is this polyribosome? So when several when several ribosomes, okay, they will attach to the mRNA. So when many ribosomes, they attach to the strand of the mRNA. So that is known as what? That is known as the polyribosomes, okay? Poly means what? Poly means many, okay? Poly means more than two, okay? So poly means many. So when many ribosomes, it adheres to the strand of the mRNA, that is known, when many ribosomes attach to the strand of mRNA, that is known as what? That is known as polyribosome, clear? Or polysomes. So the ribosome of the polysome, they translate the mRNA into proteins. Clear? So the protein is being formed from the mRNA. As I have said that the protein is being formed from the mRNA. Clear? So next thing what is present is the inclusion bodies. Okay, the inclusion bodies are what? These are nothing but they are responsible for storage of the food. Okay, so they are the reserve material in prokaryotic cells, and where they are stored? They are stored in the cytoplasm in the form of inclusion bodies. That is the reserve material, the stored food. Okay, the stored food. So they are basically present where? They are present in the cytoplasm. They are lying in the cytoplasm. Okay, so if this is a prokaryotic cell, okay, so the inclusion bodies will be lying here in the cytoplasm, and they are responsible for the storage of the food or as a reserve material, and they are not bound by any membrane system and lie freely in the cytoplasm. So as you can see, they do not have a membrane. Okay, they are very much free. Okay, they are lying freely in the cytoplasm. Okay, so they lie freely in the cytoplasm. What are this? These are the inclusion bodies. Okay, they also have the inclusion bodies. Clear? All right, yes. So the example, this phosphate granules, okay, then the cyanophycin granules and glycogen granules. All these are what? All these are the examples of the inclusion bodies. Okay, so these are the examples of inclusion bodies. That is phosphate granules, cyanophysin granules, and the glycogen granules. These are present inside the bacterial cell, and the gas vacuoles are found in the blue-green algae and purple and green photosynthetic bacteria. So in case of this cyanobacteria are they perform this photosynthesis, right? So in case of them, they have a special gas vacuole. Okay, So this is found in case of the blue-green algae or the purple and the green algae. Okay, so they are responsible for the process of photosynthesis. Okay, they are photosynthetic bacteria. So yes, this is all about it. In today's session, you have understood about the structure of the bacteria. You have seen that what is the prokaryotic cell, right? And how the prokaryotic cell is being arranged, and we have learned about the cell envelope. That is the various structures of the prokaryotic cell, that is cell envelope, plasma membrane, flagella, right? And also the pili, fimbriae. What are the various structures of the bacteria, isn't it? So I hope you have understood each and every concept of today's session. So if you like this video, do like it and also share it with your friends so that they get use of it, and be connected with Vista's learning so that you'll be getting lots of benefit out of it. I'll meet you in my next class where we'll be continuing with the next topic, that is your plasma membrane and the cell wall. So till then, take care and bye bye.